Okay, it's time to reconvene. Here's the plan. Jack Gilbert is going to be the one who kind of organizes this next session, and he will introduce the speaker. So without further ado, I turn this over to Dr. Jack.
Dr. Jack in the house. So the idea behind this session is to go a little bit beyond. I was going to give an outline that Brent already gave, so he saved me the hassle. And he did it a lot better than I could. So I think we're going to move straight into some of the presentations that really start to talk to the human interaction between this. Elizabeth, you're up first, aren't you? So you want to come up and we'll have first three talks, and then we'll have a little discussion, and then after a bio break, and then we'll have the next talks, and then we'll keep the discussion going. But if we can keep to time and have a lot of time for discussion, as I think the other talks have proven, that would be really great. Groovy.
Thanks. You want me to use this? Okay. All right, well, thanks so much for inviting me to weigh in on the skin microbiome component. And my goal today, and I think during Brent's talk, we saw that a good majority of the microbes that are coming out of the built environment are actually skin microbes, which wasn't really that surprising to me. But I'm hoping that we can, through my presentation, I'll be able to inform you more about what the skin microbiome is, why it is the way it is, and how that may transfer to our environments. So to begin with, I want to talk about some of the things that shape the microbes that live on the skin. It's an epithelial barrier surface in which the keratinocytes are continuously differentiating from the basal layer up to this top layer where they become enucleated into corneocytes. And on the surface of the skin, as well as in the invagination, such as the sebaceous gland, the hair follicle, this is the residence of the skin microbiome. Now there's compared to body temperature, the skin temperature is quite a bit cooler, and our average skin temperature is 32 to 33 degrees Celsius. So you can imagine that in addition to the fact that we're constantly sloughing off skin cells from the surface and our temperature is lower, this is going to result in a very specialized sort of niche for skin microbes to live. In addition, the topography of the skin is extremely varied in that we have areas with tons of hair, areas that are hairless. Gland density is variable. So, we have sebaceous glands, which produce the oily substances that emoliate the skin. We have sweat glands that are present in different densities across the skin, providing different levels of moisture. So some work that I did back when I was a postdoc fellow in Julie Segre's lab at NIH was to define the topographical diversity of the skin microbiome. And we looked at 20 different skin sites across different microenvironments, sebaceous, meaning oily, moist, as well as dry. And we found that depending on the site you look at, there's a huge variability in the type of microbes that are there. And for example, the areas that are very oily, such as on the face, the chest, the back, you see a lot of this blue color, which is the very dark blue is a bacteria called Propionibacterium acnes. And this is a unique bacteria in that it can metabolize some of the components of sebum, the oily substance on our skin. And then in the moist sites, we see a higher proportion of Corynebacteria, which I think we saw in some of the charts that Brent showed earlier coming out in the built environment. And the orange here is Staphylococcus, which is also a very common component of the skin microbiome, especially Staph epidermidis, which is a commensal skin bacteria, but there is also the potential for Staph aureus to reside on the skin, which is, of course, a pathogen. Taking a step back and looking at the kingdom level diversity, I think it's important to note that on the skin, and perhaps more so than other body sites, there are these fungal components of the microbiome. And there's also the viral components, and I'll talk a little bit about each of those. But this is a breakdown of of exactly how much which site is kingdom level makes up those areas. So the fungal diversity of the skin is interesting because it's not very diverse at all. It's primarily composed of this organism called Malassezia, which is genus level. At the species level, it can be a number of different species, but this Malassezia, in order to culture it, you have to add olive oil to your culture media. So it's primarily feeding off of the oily sebum on our skin. And you can see this purple on the pie charts is primarily, this is showing the dominance of Malassezia on the skin, which can, if you remember back here, it can be those orange or the red segments are showing the total fungi. So it can be a very significant component of the skin microbiome. The other notable thing about the fungal microbiome is the fact that the feet are the most diverse areas of the skin as far as fungi, and so that could perhaps be informative regarding looking at the floor surfaces where people are walking barefoot. Now, my lab's done a bit of work on the skin virome, and this is not as straightforward to analyze and to get a handle on compared to the bacterial microbiome. Even the fungal microbiome seems easy to analyze compared to the virome. But what we find is that the skin virome is lots of dark matter, and that we can't reliably classify these sequences based on to the taxonomic level, and this has to do with the fact that our reference databases are not adequately populated for this particular type of microbe. Here, this is showing relative abundance of the different viruses we found at different skin subsites. And this big blue bar means it's unclassified, meaning that we just couldn't place a taxonomical tag to that particular sequence. Otherwise, the major viruses that come out are the phage, such as Propionibacterium phage, Staphylococcus phage, Streptococcus phage, and a lot of human papilloma viruses, which is a eukaryotic virus that's been associated with different cutaneous warts and even cancers. So look, I wanted to say a few things about some of the features such as stability, and that there's been several publications that allude to the fact that perhaps you could at one time, or perhaps you could identify who's been in a certain room based on the signature that they leave in that room. And so I'll address some of the features regarding the variability of the skin microbiome. This is a figure from a paper that recently came out from Julie Segray's lab looking at skin microbiome stability over the course of around 2 years. And the thing that was most surprising is that it's actually pretty stable, which you wouldn't expect necessarily given the environmental perturbations that are constantly hitting our skin. But each of these little boxes is, each of these boxes is a person. And you can see that over time, it seems like there is quite a bit of stability, for example, in comparing T1, T2, and T3, which are the time points that were over the course of 2 years. And this is again, at a handful of different skin sites. Generally, what we find is that the sebaceous or oily sites are very stable, and it's more the drier, more exposed areas that have more variability. And that gets into interpersonal variability. How similar are we to each other, and is there really that potential where the skin microbiome can be sort of a forensic identifier? And what we find is that generally the same dominant types of bacteria are present between individuals. For example, if you look at this panel B, the back, this dark blue is again the oil-loving Propionibacterium. And generally that's a dominant bacteria in all of these individuals, but it's the remainder that's highly variable. And that makes up the interpersonal variability. Speaking more to this was a paper from Julie Segre's lab a couple years ago looking at signatures of individuality, and this recapitulated the idea that the individuality is in those low abundance type organisms that are shared across sites. For example, you couldn't find signatures of individuality in Propionibacteria or Staphylococcus, but it was these more rare type of bacteria that really we haven't heard much about, Gordonia, for example, Schuinella, or it was these signatures that were able to classify patterns of individuality. So finally, Jack wanted me to talk a little bit about what my lab has been doing, comparing different sequencing techniques in order to study the skin microbiome. This was the work of one of my grad students who just inadvertently stumbled on this project, and we thought it was really interesting and important for the community to understand that the sequencing method you can choose can have very important implications to the outcome of your study. So what we did was, and we had one swab from multiple, we took swabs from multiple body sites, and from that swab isolated whole DNA, and that same sample was subjected to 3 different kinds of sequencing, 2 of which were 16S ribosomal RNA gene sequencing. And we chose 2 different regions, the V1, V3 region, which is kind of what we were using when we used 454 sequencing. For a while, it was technically inaccessible on the MiSeq, but for this, we were, with improvements in the MiSeq sequencing technology, we're able to get this approximately 500 base pair region sequenced. And then the V4 region, which is commonly used, I think, in the environment, commonly used for gut microbiome studies. And in fact, we used this V4 region initially when I started my lab because 454 sequencing was too expensive, and we just decided that the short amplicon was better than we would have to deal with the shortcomings of a shorter amplicon in terms of its implications for characterizing the skin, which I'll get into in a little bit. And in addition to that, we performed whole metagenome shotgun sequencing and compared these 3 different methods. And first, before we did any work with the swabs, we looked at a mock community. And this mock community contained equivalent numbers of 16S ribosomal RNA genes per species, which so each species has a different number of 16S ribosomal RNA genes in their genome, so the amount of genomic DNA doesn't necessarily correspond to the number of 16S ribosomal RNA genes in that sample. So you have, looking at this heat map, you have what your actual 16S is supposed to be, that's your mock community, and what your actual whole metagenome shotgun profile is supposed to look like, and that should reflect this here, and the 16S would be even. And what we found, generally the most striking thing was that sequencing of this V4 region completely missed the Propionibacterium acnes, which you heard me mention several times when talking about the healthy skin. Whereas it then tended to overestimate the amount of Staph aureus present in the mock community. Generally, though, the whole metagenome shotgun sequencing best recapitulated the mock community relative abundance, as shown here when comparing those two. But generally, while V1, V3 region wasn't perfect for recapitulating this mock community, it was closer than the V4 primers. And as I said before, it seems like there's this huge bias against Propionibacterium when you use these V4 primers. You can see that here when comparing, while probably the best place to look is at these sebaceous sites where you have, with V1, V3 sequencing, you see a lot of this dark blue as with whole metagenome shotgun sequencing, but it's completely absent here when you're using the V4 primers. So that's important because Propionibacterium is very important for the skin. It's a huge component of the skin microbiome. But we also wanted to look at the V1, V3 region for other aspects, such as our ability to speciate, especially Staphylococcus. And Staphylococcus, we're particularly interested on the skin because you have commensal types of staph, like Staph epidermidis, but there are also types of pathogenic staph that live on the skin, such as Staph aureus. So this is looking again at the mock community. I think this is the mock community. I can't remember. Either way, you have your whole metagenome shotgun sequencing, and you can see that you have a mix of these different, no, this is not mock community because this is by skin site down here. You see you have a mix of all these different staph species on the skin. Now with V1, V3 region, you can reliably place about 60% of those sequences, those staph sequences, to the species level, although it does not perfectly recapitulate what we see with whole metagenome shotgun sequencing. In fact, it seems like it cannot place the Staphylococcus warneri, so there are some limitations to this type of placement. But with the V4 region, less than 1% of those staph sequences were speciated, and for the most part, they were incorrectly speciated in that Staph aureus came up as being one of the more dominant types in that less than 1%. So to summarize, I hope I've informed you about some of the features about the skin microbiome, why it's important to have these, this knowledge sort of at least in the back of the head when thinking about the built environment. We know that the site, the anatomical site where you sample from is very important in determining the dominant colonizer. But signatures of individuality are there, and I think there are people working on this idea of that these signatures of individuality can be used forensically. The temporal diversity is surprisingly stable, and that's despite the fact that we shower, that we use hygiene routines, antibiotics, etc. Non-bacterial members, I think for the skin at least, are very important. I did not mention the Demodex skin mites, which are a type of mite that lives in the hair follicle and have their own microbiomes probably. And there is some suggestion that these are important in some skin diseases. And finally, optimal approaches for skin microbiome characterization may be different than for other body sites or the environment. And I think we saw some hints that that might be true in Brent's presentation. I made note of the fact that one of the studies had Propionibacteria completely absent, but they had lots of Staphylococcus and Corynebacteria skin microbes in the built environment, whereas another study did have Propionibacteria. So I think that needs to be taken into consideration if you're hoping to capture the origin of some of these microbes. And so with that, this is just my general acknowledgement slide, and I will be happy to take questions. Thanks for your attention. Yes.
So we have been trying to figure out.
Well, I actually have to take off before the session ends. So if there's any pressing questions, maybe. Okay.
We're currently trying to reconcile how to integrate the use of the V4 primers that were sent out by a Sloan-funded program in attempts to unify the way that data is being produced about the microbiome of the built environment with our research on the skin microbiome, where we're now using the primer set that your group has unveiled as important to you. So do you have recommendations on what lab groups should be doing to kind of pull together data from these different primer sets? So what we're doing now is we're going back and just sequencing everything using both sets of primers, but we're unsure of how to kind of, do we analyze them as separate data sets and report them both, or how do we integrate them? So I'd love to hear your thoughts on how to deal with that.
I don't know. That's a great question, because we have V4 data from some projects, and we're not going to throw it away, obviously, but maybe Jack knows something about work going on to impute one data set from the other. So, based on V4, maybe you can make predictions about what would be in the V1, V3 sample. It'd just be nice, I think. So it's really we went back, it's really a single nucleotide in that primer, I believe, that is to blame for not picking up Propionibacteria. So maybe, but changing that single nucleotide could have huge ramification for the rest of the community, like what you captured. Then maybe you'll miss a bunch of gut bugs then, and I'm just not sure.
Yeah, we adapted the Earth Microbiome Project primers recently in order to try and accommodate an increase in some of the archaea, especially methanogenic archaea in different environments, and because we weren't hitting SAR11, the most abundant bacteria on the planet, which was a bit of an embarrassment. You can see it. You can see Propionibacterium acnes in some of our studies. It's just they're very, very low in abundance,
right.
Which is obviously wrong. They can be adapted. And we then have to do another trial. So we published a trial recently with the new V4, V5 primers, which went and compared them against all of the others and did a rerun analysis, which was published in M Systems in January. And now it looks like we probably have to do exactly the same thing with these and see if altering that has any effect upon soils and other environments. And that's the problem, as you pointed out. It's not just for human studies, but people use these for all kinds of studies. And when we're dealing with this kind of environmental analysis, we don't just need the human skin. We need the soil. We need the dog poop. We need the plants and the other bacteria that are associated with those. And that change may have other effects therein. And it's difficult, isn't it?
And I would also say, if it were feasible, I think the metagenomic shotgun sequencing is the best thing to do. But you know, the skin's a really low bioburden organ, and the built environment's even lower bioburden. It's difficult to do shotgun sequencing for those low bioburden samples. And at least, I mean, for the skin, the fungi I think are going to be so incredibly important, as well as the viruses, maybe even the mites.
So for research groups that are particularly looking at the microbiome of the hands and comparing that to the microbiome of high-touch surfaces in the built environment, what primers would you suggest using?
What did the communications person say? I'm not qualified to answer. I would use the V1, V3 primers personally, but I'm a skin centrist. So I guess it depends on your goal. If you're hoping to capture the whole skin microbiome, then maybe capturing only part of it is good enough. I don't know. Okay.
Is there someone else? Very nice talk. I have just a clarification, I guess, on directionality. So when you sample someone's heel, are you kind of assuming that the vast majority of those microbes sort of originate from the body, while some smaller portion may have been acquired in their built environments? Or are you making any assumptions on that? And if not, is there any way to explain some of the differences you see in foot microbiome, as an example, that might not be human related, but might be built environment related?
Yeah, I think that's a great point. And I always operate under the assumption that a fraction of what we see on the skin is likely to be transient, is likely to be something just captured from the environment. There are, I mean, something like Propionibacterium, staph, that's probably not captured from the environment. But we definitely see environmental microbes on the skin that would make no sense whatsoever that they live on the skin. And I think that's likely. Again, we have this issue of viable cell versus just picking up the DNA from something too. So I think that's difficult to tell if it's you can't tell if it's viable or not, and as a result, you can't really tell if it's environmental either.
Yeah. Okay. So very nice talk. So I'm very interested in your phenomena. The skin microbiome very stable. So generally, because skin microbiome, because not under stable conditions, selections, do you know why it's stable? Because there's no reason to be so stable, right?
So the stable, that's, to clarify, that wasn't my study. That was from Julie Segre's lab. I think the stability, it's not surprising. I think that the dominant organisms are stable, and it was surprising to me it was that stable because they did look at strain level of Staph epi, Propionibacterium, some of the viruses, and those continued to stay stable over those years. I think my explanation for this, and it's totally opinion, I will qualify it with that. I think what happens is when even if we use hand sanitizer, even if we scrub ourselves to death, we still harbor microbes in our hair follicles, in the sebaceous glands, in the sweat ducts. You can't sterilize those appendages, and as a result, they're sort of serving as a reservoir to continuously repopulate our skin. So that's my hypothesis about what's going. Totally, my opinion and conjecturing. So, but I think, it'd be impossible to sterilize those appendages. And
Elizabeth, can I just add to that? That's, obviously we see day to day, hour to hour variance in the relative proportions of the members, but it always bounces back. Same as in the gut, right?
Yeah.
Unless, and this is in our forensic work with Departments of Justice, this is the question I get asked most frequently when you're talking about questions you get asked, and the thing is, so if I wanted to do a burglary, how would I like exchange my microbiome for somebody else's on the skin? And no one's really done that analysis. We even know that the skin microbiome is hardly influenced by antibiotics. It shows a temporary, very transient shift, which shows no particular directional trend. Right. And it always seems to bounce back much faster than the gut. Yeah. So there is a lot of work that needs to be done there in why that is such a stable ecosystem.
Right, and so we have some work going on in the lab that's not published looking at the effect of antiseptics, antibiotics on the skin microbiome, and it is really surprising how little those interventions actually affect the skin microbiome. And I'm not sure what the implications of that are for, say, microbiome transplant or probiotic. Maybe it'll be totally ineffective on the skin for that reason.
Have you looked at the microbiomes of individuals who have very chronic skin diseases like psoriasis?
Yeah. So, my work with Janssen, we're doing a psoriasis study looking at the psoriasis microbiome during treatment with Stelara, which is a monoclonal antibody, IL-23. And this is not, so we also look at atopic dermatitis, which is a totally different skin disorder, and the changes in the microbiome are huge in atopic dermatitis, where you have these flares that are associated with tons of Staph aureus, where psoriasis, it doesn't even, I don't think the skin microbiome hardly changes with plaquing and resolution from what we've seen so far, at least. Or at least they're very, very subtle changes.
No, and are they the same as somebody who doesn't have psoriasis?
So in our study, we only have patients with psoriasis, and it's longitudinal through flare resolution. But I think Marty Blazer's group has done some work comparing healthy to psoriasis, and there's differences, but they're very subtle. Yeah. There isn't really a signature psoriasis microbiome like you might expect.
And yet there are companies out there that will say that they have come up with these
yeah, right.
Does anyone else have any more questions?
There's one back
there.
there.
One in the back. Oh, there we go.
Just a question and follow-up to your comment that the skin microbiome doesn't look very different between people with psoriasis and other people. Have you looked at function of the microbiome as opposed to the taxonomy?
No, we haven't. And that's something, it's a little tougher for psoriasis. On skin, the top layer of skin is enucleated, so when you take a swab, you get minimal contamination with human cells. But a psoriasis plaque is sort of hyper-differentiated and hyperkeratin. It's really scaly, and the cells aren't enucleated, so it presents this challenge where you're trying to do shotgun sequencing on the microbes, but you have to deal with the 99% human. But I think there's people probably doing that, I imagine.
Yeah, I think my comment was not meant to be terribly specific, but it's more a comment that you can look at the taxonomy of the communities, and perhaps it doesn't tell you everything about whether the function is the same.
Yeah.
One of the arguments in that would be that you'd have to do transcriptomics.
Yeah.
To actually get at that. Any more? Oh, yeah.
No, she's going to have the last word. So I've been very interested in your work ever since the Grice and Segre paper. because I'm interested in how the indoor environment influences the human microbiome. And as Julia explained to me, the skin is the largest organ.
That's actually not true.
Well, I said it wasn't true also, but I accepted. The lung actually has more surface area than...
And the liver's heavier.
All right, so we use different criteria for what's
But either way, the skin is not the largest organ, though
Doesn't
that's
matter.
perpetuated.
The question I wanted to ask you is, so as I read your work, the oiliness and the wettedness of the skin were the major determinants.
Correct.
And I was interested because we know a lot about how the indoor environment influences human perception of thermal comfort. Mm-hmm. Those of us that have been involved in the ASHRAE community. And so the skin temperature isn't the same all over the body either.
No.
And the skin is the major regulator of our core body temperature. And so it responds as the ambient temperature around it goes up or down. And I just wondered if you've looked at all, or if you know anything at all about the influence of temperature as well as moisture and oiliness. And then focusing on moisture and oiliness, what about other chemicals that may be present, cosmetics, mm-hmm. soaps, and so on, and how those things that we have some control over influence what's on the skin?
Right, that's a great question. And I'm not aware of anything temperature specific, though, you know, I would hypothesize that someone that lived in a very warm climate, there may be differences between that and someone that lives in a cold climate. But yes, so one in my first slide, it showed the difference in body temperature across the skin. And yeah, and I think we need to take into account the products we use on our skin. Peter Dorstein and Rob Knight, Rich Gallo had a very nice paper recently looking at mass spec across the skin surface and along with 16S sequencing, and they found that they could tell like who used what shampoo and soap based on the mass spec data that came out. There were those products still that were persisting on the skin. So
they hadn't washed for like 2 weeks as well.
Yeah, exactly. And so, but those things were still there. Yeah, it was 2 weeks.
A man and a woman as well. It was Peter and his wife for the IRB buffs out there.
Oh, it was them? I learned something.
But at least they did it together, right? So.
Wait, I'm thinking, how much do you have to pay someone to do that? They did it voluntarily.
They couldn't get anyone to do it voluntarily, so they did it themselves. All right. Yeah, we have to move on. So, do you want to come up and load up your slides? And if you want to grab her now, go and grab her now. Oh, unless you're...
I was just going to say that the skin is a barrier, but an imperfect one, and chemicals that applied will go across it in both directions, not at the same rate, but finding something 2 weeks later is not exactly surprising.